Our next expert is Dr. Snezana Maljevic. And Snez, as we will call her from henceforth, is originally from Belgrade, Serbia. She spent a lot of time in Germany doing her work there. And because we're the lucky country and we believe in immigration, I'll let that sit. Snez is going to talk to us about brain in a dish, modeling epileptic encephalopathy using human stem cells. So, winning the prize for best title, Brain in a Dish. Take it away, Snez.
Okay, thank you very much. Not an easy task after wonderful talks from Anne and Ingrid. But I just want to say that brain in a dish is not so original in scientific world. We use it very often. But this topic and what I'm working on has really changed with when I got involved with epileptic encephalopathies. So I did my PhD on KCNQ channels, and obviously KCNQ seems to be a very popular channel here today. And when I did that, the KCNQ was only causing benign form of epilepsy, so we only knew that it causes epilepsy that appears in the first few days of life, but most of these kids would be fine even without treatment. So that was a nice intellectual challenge, like, yeah, let's use this as a model of epilepsy and maybe we can help other severe cases. And only many years after, and actually only a few years ago, 6 years ago, came that famous paper with epileptic encephalopathy mutations in KCNQ2. And suddenly it became really important and urgent to study KCNQ. And it was also because of my previous work that Sarah Verkuijsen reached out and we did the first functional analysis. You will see a bit of that here. But it was then something that led to involvement with parents, and Peter, who is now online, is actually a parent who contributed iPS cells or cells from his child to develop iPS model in our lab in Germany. So it just became much more connected to your stories, and we were much more exposed, and I think this is how the work now goes here. So I'm really happy to be here. And I kind of tried to make it not like a very complex cloud you see on the screen now, but just to answer questions that we normally get from people who would like to use or who have heard of stem cells and would like to get involved in modeling encephalopathy their child has. And the questions we asked were, why do we need stem cell models, or do we need them at all? Then maybe also for some of you, what is it? What does that mean to use the stem cells? And then we'll look at some examples of what have we achieved at the moment and where we would like to go, so what we hope to achieve. And I kind of went with sort of a parent's guide to stem cell modeling, just like kind of telling you how much time it can take and what it does involve. So let's get on with it. Why? So, you know, and you've already heard, the talk is mainly, or I work mainly on genetic epilepsy, and I think because everybody here has their genes, it is all okay. We all know some mutation will start the whole process. And having identified or diagnosed mutation often leads to the next step, which is functional analysis. So most of the time, and now using new bioinformatic tools, we could predict whether mutation will be pathogenic or not. You will also know that if parents don't have it and the child has it, so it arose de novo, that could also mean that the mutation could be pathogenic. But if you really want a final proof, you would then try to express that mutation or try to analyze it in a certain way and just see if it does change the function of the affected protein compared to the one that does not carry the mutation. And then if we understand the pathophysiology, we always hope there is a way to target that pathophysiological mechanism and develop treatments. So for that, all that, we need disease models, and you've seen already the cute fish this morning, but I'm going to talk about my favorite models, and they start with frogs. So we use, often at the beginning and for fast screening of mutations that are identified, we use frog eggs. So the frog has many eggs in their stomach. You can take them out and you can use them as a cell that will kind of be a little incubator for the proteins you're interested in. And for instance, for KCNQ channels, for some of the other channels it works really well, GABA receptors. So it just is a very neat and fast way to test whether the mutation changes the activity or the function of the channel. You also see some little cells next to it, and these are just the regular mammalian cell cultures that we also use to just express mutations and basically just compare, does the variant that is introduced cause a change compared to the protein that doesn't carry this variant? So can we identify that difference? Sometimes it's easy and fast, sometimes it takes a long time, but most of the time we would get the criticism that, you know, what does a frog egg have to do with the human brain? And we, of course, don't claim it does, but it does remove all the other interfering molecules that could actually mask the effect that we want to see. But it's just the beginning of the story. Of course, if we want to look in more detail, we have to provide some neuronal environment. And for that, we use a lot animal models, and I actually here also emphasize mouse models because this is what we work more. We can use those models to extract neurons, to analyze them in dishes. We can express in those neurons our proteins and see how they affect the firing. Or we can just wait some months and try to generate a genetic model and then analyze it. And you see that this little mouse carries a bit of a funny head. That is the EEG system, so it is recording the activity. And then recently, let's say in the last 5 to 6 years, everybody started working on models that are using stem cells. And when I say stem cells, I actually mean induced pluripotent stem cells, because the other ones are more complicated and more ethically difficult to use. So before going into that story, just for the KCNQ2 lovers, to show that there are some there is some information that is already found in a very simple models. So these are, for instance, recordings from oocytes. So you just have a frog egg, you express the wild type, which is the big the lines that are there. I'm sorry, my screen is not working, so I can't show you. But you see that this kind of activating current, where it says KV7.2, this means this is a normal current. This is how this channel would work. This was what we would record in an oocyte. And all the other three are the three variants that are found in children with epileptic encephalopathy. And you see that they don't have any currents. And you even see that if you look at those bars on the other side, that not only they don't work on their own, but if they kind of assemble with the healthy channels that are still present, because every child would carry one mutant and one healthy allele, they impair the function of the healthy allele too. And this is how we kind of identify that these mutants actually cause a dominant negative effect, and that is why the situation in these children, so the disease can be more severe than the ones that we studied 15 years ago with benign syndrome. And yeah, this is the frog, not the original one. And this is the system. I just want to say, why is this analysis fast? Because we can use automated systems. So we can use plates and have many oocytes recorded in parallel. And this procedure is then relatively short, especially compared to what you're going to see when you come to mice and stem cells. One of the things for precision medicine, and I think it came really nice in this study, is that we could actually differentiate which of the mutants, which of the disease mutants would be responsive to retigabine. So you see here that wherever there is a difference between two graphs, that means this is a shift caused by retigabine that would be actually beneficial for the patient. But for one mutation, it doesn't happen. So for that one, we probably wouldn't advise if somebody asked to try it at all. So frog is maybe really simple, but it does provide some information that can be used, even for precision medicine purposes. Of course, genetic mouse models are the next step and a very useful step, and I don't know if there are going to be talks that will explore them more today, but I think they would take the whole symposium to address. There are many genetic models that were developed in recent years, especially because of that famous technique that was mentioned today, CRISPR-Cas, because now generating mouse models became also faster and easier. The process and the procedure is still pretty long. We do have to address the whole behavior and EEG, and then analyze the electrophysiology, so use the brain slices, do the test of drugs. So it is like still a long and yeah, painful process sometimes, but it does provide a lot of information. We really learned a lot about epilepsy and the mechanisms of epilepsy from mouse models. However, we know also, and this is what is mentioned very often in recent times, that mouse models do not truthfully represent human biology, how surprising, but that clinical studies that were often done and very successful in mouse models do not translate so well in phases 2 and 3. So this is where many drugs fail. And one of the reasons is that we are, of course, different to mice. And this difference, and it has always been a question, can we then just like do something more in human cells? And of course we don't have access to brain tissue as easily. And that is why the next technique, or the technique that appeared and that uses stem cells, became really important. So stem cells. What is stem cell? I kind of thought how to define it the easiest way, and I think stem cell is just a cell that has many options. So it is a cell that can become a neuron, a gut, a blood cell, whatever it wants. And we all have, start with them. So during embryonic development, you will see that in this inner mass of cells that are in the blastocyst, each of them can turn into any tissue of the body. And while this has been explored for many years for the use for differentiation and for use for treatment, it is ethically really complicated, and it is also really complicated to do the experiments. You've also heard about adult stem cells. Those are those that are repairing and that people like to also explore a lot. But I think the whole thing changed in 2006 with Nobel Prize 6 years later with the induction of pluripotent stem cells. And what this means is that we are actually now able to make the cell, for instance, skin cell or a blood cell, forget the decision it made a long time ago, go back and just become again a cell with options, become again a pluripotent cell. And if you wonder how that process, which we call reprogramming, happens, or what it requires, and you thought of a cocktail, you're right, because we give them now a whole menu of cocktails in the meantime. This procedure to turn or reprogram a somatic cell and turn it back into a stem cell has now really developed as a kind of relatively easy method. You can just order that in a facility. It will be done within a month or 2 or 3. But that's a relatively easy procedure. And it's kind of getting better and better and faster. So we do use those. And we have, reprogramming has helped us develop a whole new paradigm. So we basically now can start from any of the somatic cells that are easily accessible. We in the lab love fibroblasts, and I know that involves biopsies. But one of the reasons for that is that they're the easiest to keep. So to keep in culture, to keep in the biobank, and to reprogram, they're kind of the most stable of all the others and give the fastest results. But in the meantime, they're also very nice reprogramming, successful reprogramming projects with keratinocytes, so hair follicles, or with blood cells, which is probably the easiest for everybody. After the reprogramming, these cells are then called induced pluripotent stem cells, and we can differentiate those in neurons. And one of the options would be, of course, to put them back in the brain and fix it in a way, if we somehow fixed what is going on. But we're not, I don't think that epilepsy is going to profit from that very soon. But what we would like to use it for in genetic epilepsy is disease modeling, so kind of understanding in a more human setting and more native setting, what do the mutations that are found in epilepsy do in this brain in a dish. So we said we have patient, we have reprogramming, and then we have pluripotent stem cells. And one of the things that we would like to do is we can, of course, compare the properties of that cell that we took from patient or made from patient cells to the general population. But we can, and then there will be some variability. It depends also when you take the skin punch or with the skin biopsy from? How old is the person? What is the background? On what day it happened? I guess many things that can influence how well is your control going to be a control for your mutation. Which is why we decided to use the CRISPR-Cas, also already meshed scissors. I have a slide on them, I think. And just fix that mutation. So if the epilepsy is known to be caused just by this one single nucleotide exchange, we're just going to go into those pluripotent stem cells and change that mutation, turn it back to what it's supposed to be, and generate in such a way our control line. And then compare that or use that for disease modeling. So the editing is this what Anne already mentioned, but I just kind of think picture is always nice. You see the double strand of DNA, and you see that we can, with this sgRNA, which is really specific for the sequence we want to fix, we can just target the sequence where the mutation is found, and then with oligos that are on the right side, we can use that to basically insert unchanged or healthy allele again. So once we have those, these are how the iPS cells look like. They're kind of nice and healthy, I would say. Not here they don't have feeders, so if you have looked at some pictures where they look more round, this is because they're growing on fibroblasts. These are just grown on matrigel, so they look kind of more like a freestyle colonies. And what you see below are the pluripotency markers. This is how you characterize that they actually now have all the possibilities to become any type of cell we want them to be. And this is part of the differentiation process. And because we speak about epilepsy, our differentiation protocols are actually generating cortical neurons. And now you see that one of the neurons has a little X that is the mutation. And then we also generated the one that doesn't, so the corrected one. And now we can compare the properties of these neurons. So how does the differentiation happen? There are several different protocols, or many different protocols. So it's something like a cooking recipe. Everybody does it in their own way in the meantime, but there are a few main strategies that you can use. One is more native, so basically using small molecules to mimic what is happening in brain during development. But that one is probably nicer for the development of neurons that will be more similar to what we have in our brain, but it does last longer, and it's more variable what you're getting in the end. The second method that we also explore a lot is using the overexpression of one of the neuronal genes, and that can turn the iPS cell into neuron within a week, which is amazing. So I'll just show you how that all looks like. So you see iPS clone that it all starts from. Then you can go over embryonic bodies that looks a bit like a native development. Then we can have spheres and differentiated neurons, and everything that is in green on the pictures below are neurons in these cultures. And some red are here younger neuronal precursors, or they're some of the glia cells. And how they look when you use the FAST protocol, this pretty picture from Ben Rollo in our lab, you see that within 6 days you're getting neuron-like cells. So those cute guys over there would be neurons. They wouldn't still fire and be functional after 1 week, but another week and they'll be fine. So we came here to the stage where we want to do the characterization. So what can we do with those neurons we generated? We can look at the properties of single neurons, or we can look at the properties of whole networks, or we can look at the expression pattern, or we can test drugs. So this is what we kind of want to use them for. I just show you a few examples. I don't go into any mutations because we still have to finish those studies, and I didn't want to overwhelm everybody, but what is shown here in blue and green are the neurons that are firing, and that these neurons were obtained with these two different protocols. So the green one is the one that is obtained with a 6-month long protocol. So basically these neurons were grown for 4 to 6 months before the recordings were made. And you see that they fire kind of more and higher action potential. And the blue ones, these are the ones that you can generate within a week and measure after 2 weeks. they're still firing, but they fire just less sexual potential. They're a bit younger neurons. That can still be enough for us to model disease. Concerning the network activity, you also heard that we use multi-electrode arrays. So they can be helpful as in fish. So basically you are growing cells on the top of the dish that has electrodes embedded. And you can record then the activity of neurons. Basically every single spike you see here, every single line you see here is a line that shows the activity of, I don't know what's going on, yes. Shows the one single spike, so one single action potential of neuron. And then when they come together, like in the picture on the right, those would be bursts. So those would be something like a more mature activity. This is when neurons start talking to each other. And to obtain such activity in cultures, we need to grow these neurons together with glia cells. So it needs more optimization and more time probably to also generate them to be nice and regular. But it's possible, and this is the signature that we would normally look at also in epilepsy models. So in mouse models, it's just the neurons that you extract from a mouse model would behave in the same way. So, let's see what happens. Okay, so I said, like, if we want to kind of define what do we want from our models or in vitro models that are kind of good models, we would like to have reproducibility, and this has been a problem for a long time. Anybody who has worked with any kind of iPS cells know that the lines that you generate from the same patient can be pretty different. But we are getting better there. I think we're getting better protocols, we're optimizing more, and we are kind of establishing those protocols always that we have, you know, enough number of controls, enough number of patient lines so that we can reproduce that. Scalability is probably not a problem. As you know, one of the characteristics of stem cells is that you can multiply them and you can keep them and you can grow your biobank, and you would just have always a source of material to study. You can differentiate both neuronal cells and glia cells if you want to combine them, or you can combine them with different types of glia cells. So these protocols are now giving practically all we need. Maturation is still a bit of a tricky thing, because I don't show you here how the firing rate in mouse neuron would look like, but believe me, it would be higher and better and tighter. So they would fire, you know, adult mouse neurons, and our neurons, they do fire more. So what we are getting are probably young stages of neurons. I think for epileptic encephalopathy, that is not bad, because actually the neurons are, the young neurons are the ones that are affected because disease expresses so early. But we would like to have the chance to also achieve that maturity within less time than 6 months. Then we look at the connectivity. So I already said neurons should not only sit there by themselves in our brain, they talk to each other. So we want them to talk to each other in a dish. And we can help that. I think we are developing also protocols for that. We are adding some glia cells. We are growing them in a tighter constellation, or we are growing them in 3D cultures, as I'm going to just show you. And that all is also related to the developmental processes. You would kind of wonder, especially for developmental encephalopathies, can we also see what is actually happening during development in these neurons? That if you grow the culture just in a dish, like you know, as the neurons you've seen, you wonder, would you be able to see the migration, the proliferation, and all the other processes? And would you be able to see that with a protocol that is just overexpression of one protein? So probably not. And I think in the recent years, many people talk and have probably read a lot about different organoids. So like generating small pieces of tissue that can sometimes be really useful, like in the case of liver up there, or eye, or heart. But talking about brain, it's always tricky, because nobody expects to grow a brain in a dish that will exchange a brain of a person. So we want to use the brain, or brainoids, more, again, as an experimental model, a model that will help us look at some of the processes from disease. This is kind of a busy slide, but I just want to show you that there are also for neurological disorders many approaches that have been developed and that have been tested. And basically, they're all based on growing a little balls of neuronal tissue for a long time in culture with certain soup around them, trying to reproduce what is happening in brain. And it's possible now to make actually small brainoids of different brain parts. So you can make the one that is just the cortex, or you can make the one that is hypothalamus. So you can kind of now walk through the brain and just like using specific soup growing around those parts or in those parts of brain generate some neuronal structure, three-dimensional structure that are actually reproducing what is happening in the brain. So we started doing one of those protocols in our lab. This is how the brainoids would look like. So it's not like a real or small brain, just like a little ball, as I said. But it does take a time to develop. So you see that the whole process that takes at least 80 days before you come to the, or 90 days to come to the point where you would analyze them. Before that, they're just like growing and experiencing all the changes. What the below picture would actually show, or what I want to show with the picture below, is that you're getting all the cortical markers. So you kind of, these structures tend to assemble in a way the brain would assemble. So you are getting different layers, and it is possible to test and compare those layers between the organoid that you get from mutation and the one that you get when you correct the mutation. And I think this is going to be a really helpful approach for developmental processes and looking into them. So, yes, when people ask how and what can they expect, if do we want a biopsy, and should they do that, and we get approached by those questions often. The process is simple, right? But it's just not easy, and it's not short process. It does involve, and I was just talking to, I don't know, Janus, to somebody who was sending cells. Yes, hi. So it does involve taking a biopsy or taking blood cells. It is not, biopsies are not that invasive, but are invasive. So in some situations, people would decide not to do that, so rather opt for blood or some other somatic cell line. It does take, especially to transferring those cells within Australia and also from outside of Australia, is a bit of a challenge, but we're kind of managing that at the moment. And then the reprogramming, as I said, is kind of established protocol, so that can be done relatively easy. Gene correction is not as established protocol because it depends where mutation is, which gene is affected, so sometimes it is a trickier change to make. But all of those, cost money. So it is kind of a question whether you sometimes just pull a nice pool of controls and then compare the mutations to the controls, or you do the correction each time. This we are still trying to figure out. Then the whole process of differentiation and analysis can take a long time, but we do hope that those times will shorten and that we will be able to produce more reproducible assays and faster assays that will then also be useful for more drug screening. We can apply now, of course, drugs on the cells that are as active as we have them here, and they would sometimes show a nice difference, but we want them to be more neuron-like, so that we want our in vitro assays to really represent what is happening in the brain. And I think it's on a good way. So, yes. I thank you. I'm happy to take any questions.
Anybody any questions? Thank you. We've got microphones going around for questions. This one at the back.
Hi. I just have a couple of quick questions, hopefully. I was just wondering how far away you are from clinical trials for children with encephalopathies as a treatment for epilepsy. And also, there's a growing trend for athletes and stuff to use stem cells for injuries. Just wondering if there's anything being progressed about using stem cells for children with severe epilepsy around the other things like movement disorders or injuries or like kidney damage, liver damage, things like that, if stem cells have a role to play in that sort of treatment?
So to both questions, I mean, I don't think it is at the moment feasible to plan transplanting neurons to treat epilepsy, because we're just not there to really understand what goes on and how well connected the neurons we transplant will become. But we are understanding more and more, and I don't exclude at one point, you know, it's going to be a procedure that people would go for, opt for. Concerning, I know for sports people, but just like whenever we talk about stem cells that are currently used, it's all peripheral, it's all like, you know, it's your knee that you can treat. So it's not your head and it's not your brain. So I just don't think that it's at the moment there. But I do think that there are ways to, you know, that this will help us more understand what happens in the brain so that we can develop a therapy that will be more efficient. I think that is more where we are heading at the moment. I mean, I know that there are people developing inhibitory neuron stem cell-based planning transplantation, but I just don't know how feasible it is. I don't see it very close at the moment.
Thank you.
Snez, when you say therapy, you say people are developing a therapy. What do you mean by therapy? Do you mean medicine, medical treatment, or what do you mean by that word?
So I mean all types of therapies. So I mean, you know, using this as a readout for small molecules for repurposing before you start, you know, trialing drugs on a child is probably something that could be useful. Steve will talk later about molecular therapies. So it is also a very interesting and probably a very good opportunity for development of treatment. So I think, yeah, all kinds of therapy, but I think it's just a good readout, and we have a human background that we lack in the mouse or other models, and it's kind of just, I don't advocate we are replacing those though, because we are not there yet, but it's like a complementary approach.
All right, any other questions? And then we have one over here. Yes?
Sorry, I just wanted to ask, were you looking for more human cells to sort of find out what's happening with the different diseases? Is that what you're sort of hoping for? So you're asking for kids with the volunteers to
well, you know, we kind of have already enough volunteers. So, you know, like we are just... sometimes we're approached by people who would like to have the stem cell-based research on their, you know, like on the mutation that their child carries. This happens, but we also kind of, because we have pools of different mutations within genes that we are studying, so we're kind of collecting those, and I think those would be also useful. If there is any new therapy, maybe you would like to run through the pool of mutations affecting certain gene before you go for, you know, clinical studies. So in that sense, yeah, we are interested in collecting more biomaterials.
All right. Oh, yes, we have one here. Pardon, yes? From Anne?
Thanks so much. I just wanted to follow up on some of the questions too, just in the sense that we learn so much from each other, and I think it's quite amazing that the work that you were doing earlier, really without an eye to this population, is so applicable here. I think in the same way, you know, we can learn so much from other fields where potassium channels are active, and so I think it's worth the families knowing there's a whole world of potassium channel work being done by our colleagues studying Lou Gehrig's disease, the farthest thing you might think from pediatric epilepsy, but quite a lot of applicability. And it turns out that while we're talking about a rare epilepsy, Lou Gehrig's disease is not as rare, and so drugs that we find might help them, drugs that they find might help us, and there's really an opportunity for synergy when you find the right drug in the dish. It might not be so easy to get companies to market or even just to fund a trial just for us, but they might be willing to for the other population, and I think that's important for you all to know that we're looking for ways to make this happen, even if it's not just within our own groups.
So I think that's an incredibly important point, because basically what you're saying is that our cousins if we continue to use that metaphor, exists not only within the genetic epilepsy community, but more broadly with other disorders and diseases which share a channel or a piece of human DNA that isn't working properly, but might not have seizures, might not have epilepsy at all. Do I have that correctly? All right, so that's an incredibly important point. We should probably wrap then because we will, again, have an opportunity at the end to do a roundtable of questions. I just want a little plug for our international visitors. This is Haig's chocolate. It is worth coming to Australia. And this is from South Australia, our friends in Adelaide, Tanya and all. So Haig's chocolate, going home with the Americans, going home with the international, and staying right here. And if there's any left, I'm eating it. Here you go. Congratulations. Thank you very much.